My name is Pooran Dewari. I'm a postdoc in Dan McQueen's lab at Roslin Institute. And I've got some pictures for you from Roslin. So on the left is the Roslin Institute, which is located in this beautiful Scottish countryside. By the way, the purple flower in the foreground, that's thistle, that's the national flower of Scotland. And Roslin being the birthplace of Dolly, I thought I'll share some pictures of Dolly's friends from Pentland Hills as well. So my job today is to walk you through some basic steps of my favorite molecular biology technique, that's chip sequencing. I know in these kind of courses, everyone is more excited about the practicals, and these kind of theory lectures could be a little bit boring. So I'll try to keep it to 20-25 minutes. And meanwhile, during the talk, if you have any questions, please feel free to stop me and ask questions. So the main idea is to bring everyone, bioinformatician as well as the wet lab biologist, to kind of bring on the same page for tomorrow's practical course. So I'll give you a brief introduction about how we prepare libraries and also discuss some of the key papers, just to give you a flavor of how we use ChIP-seq data sets to understand gene regulatory mechanisms and other biological processes. Okay, so I'm aware that this is a little too complex for opening slide, but I intentionally chose this one to give you a sense of how complex the chromatin is. So often when we analyze ChIP-seq data sets, we are kind of thinking in a very limited view of just a couple of proteins we are working with. But in reality, chromatin structure is highly, highly complex. So just don't worry about the details in here. Just remember, we are starting off with a silent chromatin state on the top. And through a series of chromatin remodeling, this silent chromatin is kind of opened up and different machineries are recruited, leading to activation of the gene. So chromatin remodeling starts with the recruitment of different chromatin remodelers and histone acetyl transferases at upstream activator sequences just before the promoter. These chromatin remodelers and histone acetyl transferases, they kind of embellish the nucleosomes with different histone modifications, particularly histone acetylation, leading to histone eviction, thereby exposing the promoter element so that Pol2 and other transcriptional machinery can be positioned. So there's a lot happening here, but how do we know these events? How do we know the sequence of these events? So that's where ChIP-seq kind of comes into picture. So ChIP-seq captures a snapshot of this dynamics. So think of ChIP-seq as a frame in a movie, and by putting these frames together, we get to know how these different events are kind of morphing from one phase to another, eventually leading to the activation of this particular gene. So if I could break down chip sequencing into 4 steps, we start a ChIP-seq experiment with the chromatin or cross-linked cells, where you have chromosomes with lots of chromatin modifying enzymes and histone modifications onto the chromatin. In this case, let's say you are interested in this particular transcription factor. So we cross-link the cells to make sure the DNA protein interactions are stabilized to avoid artifactual rearrangements during the processing. Next, we fragment the chromatin. So the shearing of chromatin, that's the most crucial step, and that takes much of the optimization in chip sequencing experiment. So you chop the chromatin into smaller fragments, and obviously you will have some fragments with your transcription factor bound onto them, others won't have them. And then using a transcription factor specific antibody or any antibody that's specific to your protein of interest, you pull down these chromatin fragments. So this step is called chromatin immunoprecipitation. So you are enriching your transcription factor bound fragments in this case. You'll also get some background sequences. And then eventually you make library, purify the DNA, make library, and send it for sequencing. And then using bioinformatics approach, we align those reads back into the reference genome or the chromatin or the DNA sequence here. And when you pile up the sequences, because we are enriching for these transcription factor bound sequences, you would see peaks for each of these sites. So that's essentially what the chip sequencing workflow looks like. Of course, there are each of these steps are much more complicated, and the optimization and development of the chip sequencing, it took almost 25, 30 years. So if we just have a look at the history of the chip-seq technology, in its very early phase, chromatin immunoprecipitation, the idea of chromatin immunoprecipitation has its roots in back in 1980s. So this paper in 1984, they looked at the occupancy of RNA polymerase at active genes, and they used protein DNA cross-linking using UV light. Although UV light is a better cross-linker for RNA protein cross-linking, but nevertheless it works. And 4 years later, formaldehyde was introduced for cross-linking protein DNA cross-linking, and this is a reversible cross-linker. Nothing much happened really for the next 10, 12 years, except that real-time qPCR was introduced in 1993. So before that, for the visualization of the enriched DNA sequence, people used to do southern blotting and playing around with radioactive probes, but real-time qPCR made life much easier. So, and it's also more precise. But the real game changer in chip sequencing was combining chip with microarray, which is called chip on chip. So the first paper came up in 2000, and this was a big deal. So in just one single experiment, this paper profiled Gal4 activator across the entire yeast genome. So as seductive as it sounds, microarray has its own limitations. So for example, you can only get information that's already there on the array. So there's no de novo information. You can't do multiplexing. So that was a good timing because Solexa, the next generation sequencing company, was founded in Cambridge University. I think that was back in 1998, but the technology took like a couple of years to develop. By the way, Solexa was later acquired by Illumina, but that's another story. So when it was combined with the next generation sequencing, Solexa sequencing, that was actually the breakthrough in chip sequencing, and in a way, the first chip-seq paper. And in just one paper, they mapped 20 histone marks across human genome, RNA polymerase to CTCF, and there's a lot of information in that paper. And I'll briefly touch upon some of the key results from that paper. So the main idea with chip-seq is that it's a single nucleotide resolution. You could do multiplexing. And in terms of the major development, the last major development in chip-seq was back in 2015 when chipmentation was introduced. So this makes chip-seq much more easier, shorter, and low input requirement. So you could do chip-seq with as little as 10,000 cells. I'll discuss this paper as well later. So in terms of chip-seq workflow, here's the overview. So as I mentioned earlier, you start with cross-linked DNA protein interactions with 1% formaldehyde. For most of the experiment, we go for 5 to 10 minutes. And the most crucial step is chromatin shearing. So we target for 200 to 600 base pair fragments. So this chromatin shearing makes sure that the chromatin fragments are kind of solubilized easily. And also you don't want to pull down the entire chromosome. So you will lose the resolution if you don't fragment the chromatin. And recently there have been like lots of advancement in terms of chromatin shearing protocols. There are now water baths, sonicators available where you could process multiple samples, and the reproducibility between samples is really good. So once you do the chromatin shearing, you keep a part of it, which is called input. So that gives you kind of a background for the chip-seq tracks, and the rest of it goes into chromatin immunoprecipitation. So you use antibodies specific to either your transcription factor or histone modifications or histone stem cells or any other histone acetyltransferase or chromatin binding protein. So you enrich the fragments, pull down the chromatin fragments, and then there's some cleanup, reverse cross-linking, purifying DNA, and then the library preparation. So you add adapters so that it's easy to sequence them, and also you amplify the library. And the great thing about the library preparation is that you could use different set of barcodes so that you could multiplex different libraries and sequence them together. So, and then eventually you send them for sequencing, and then the fun part is the bioinformatics analysis. So here is some real life example of a chromatin sharing optimization from our own lab. So this is for the Atlantic salmon embryo that we recently prepared some ChIP-seq libraries. So as I told you earlier, the main idea is to chop the chromatin to 200 to 600 base pair size. So the tape station profile for the same here. And once you have immunoprecipitated the samples, it's a good idea to validate chip samples because it's a very expensive business, very costly business. So we validate chip samples at positive and negative controls regions. So it's a good idea to kind of keep 2 positive and 2 negative control regions. For example, in this, if for these 3 libraries, so you have K4 trimethylation, K27 trimethylation, and K27 acetylation, some active and some repressed mark. And we validated these chip samples at one active locus, that's a Tata binding protein promoter, and one repressed promoter. So as expected, the positive histone marks, the activating histone marks, they'll be enriched at the Tata binding protein promoters, so in blue and purple, whereas at the repressed promoter, you would see enriched levels of K27 trimethylation, which is a repressed mark. So it's a good idea, but sometimes you'll be working with a not very common model organism, for example, in aquaculture. So a good idea is to kind of go for a low pass or pilot ChIP-seq first to get to know some of the common well-known sites in the genome, and then design primers for the later on chip validations. IgG isotype chip is also used just to get an idea of the background pull down. It's not, so input is the most preferred input control. With the IgG isotype immunoprecipitation, sometimes we don't get enough DNA for library preparation, but it's very useful for qPCR validation. Okay, so the first paper, ChIP-seq, that came up in 2007. So this one profiled 20 histone methylation marks in the human genome, histone variant H2AZ, and then RNA pol2 and CTCF. So this was the time when next generation sequencing was being developed. So 36 base pair, that was the read length, and the typical output for this machine was 1 GB. So you can, I mean, we have made a lot of progress in sequencing technology as well. So nowadays with NovaSeq, we can get up to 3,000 times more output than this first generation Solexa technology. So there's a lot to take in this paper, but I thought I'll just share just one figure, which is relevant to gene regulatory mechanisms and kind of simple examples. So here they profiled pol2 at 4 different categories of genes. So genes that are expressed at high, medium, low, and silent genes. So they grouped 20,000 genes, human genes, into these 4 categories based on their gene expression. So as expected, you would see high level of pol2 occupancy at highly active genes and really low expression at silent genes. So that was a bit surprising because you would expect there will be no pol2 at silent genes, but as we'll discuss later, there are some genes which are kind of primed or poised for activation. S3K4 trimethylation histone mark, it positively correlates with gene occupancy, gene expression. So it looks similar to pol2, except that you see a dip at transcription start site because it's a nucleosome-free region. So there's nothing in there, but there's a really nice nucleosomal kind of banding pattern around the transcription start site. S3K27 trimethylation, on the other hand, is a repressive mark. So it negatively correlates with the gene expression. So the silent genes, they have high levels of K27 trimethylation, and the active genes, they have almost no K27 trimethylation mark. So this was kind of, I think most of these informations were already available based on chip-on-chip microchip microarray experiments, but it kind of gives you a one base pair resolution of these, and also it discusses some other histone modifications that were not explored earlier, but I'm not discussing them here. But and then then another paper came from the same lab, follow-up paper 1 year later, and in this paper they looked at histone acetylation mark, so 18 of the histone acetylation mark. I know there's a lot to take in this heat map here, but just focus on the right bottom of this figure. So this big block here, that shows that all these histone acetylation marks, they are co-localizing on the same sites. And all of these are active genes as seen by the Pol2 occupancy. So what it says that these histone modifications, particularly the acetylation and some of the K4 trimethylation marks, they are kind of co-localizing and they are working in a cooperative fashion to prepare chromatin. So it's just not one single modification that drives histone activation. It's always a cooperativity between different histone modifications. And as you would expect, the repressive marks on the top, shown on the top in this heat map, these are absent at these active genes. And some of the key marks that we use nowadays, some of the well-established mark for activity, transcriptional activity, they are shown here with arrows. So K27 acetylation and S3K4 trimethylation, they will be present at active genes, whereas K27 trimethylation will be absent. So based on these and many other papers, the general consensus we have now about histone modifications at transcriptional regions is that we can categorize promoters into 3 different categories. So active, poised, and inactive promoters. So just by looking at histone modifications, we can predict transcriptional activity. So the active genes, they have RNA polymerase II sitting at the transcription start site at the promoter site. And you have the nucleosomes, they are kind of decorated with H3K4 trimethylation. So that's the active mark. Inactive genes, on the other hand, obviously there is very low or no RNA Pol2 occupancy. And the nucleosomes, they are embellished with these repressive mark, most notably H3K27 trimethylation. What's really interesting is that some of the promoters, they are kind of kept in this poised or primed state. So these are called bivalent chromatin modifications. And these contain contrasting or opposing histone modifications. So as you can see, the active mark S3K4 trimethylation and K27 trimethylation, the repressive mark. And these kind of poised promoters are really important for developmentally regulated genes, for example, transcription factors that are required for differentiation of stem cells or during embryonic development. So when cells need to rapidly activate certain genes, they just erase the K27 trimethylation and the active mark being already there. So it's a rapid increase in activation of the chromatin of these genes. So that's the story about the promoters. So another important gene regulatory sequences are enhancers. I think in the post-genomic era, the main challenge is to validate these gene regulatory elements. So enhancers, by definition, these are cis regulatory sequences. So they're on the same DNA sequence that's on the same chromosome, and they enhance the expression of target genes by kind of forming these loops. So mostly enhancers are kilobases away from a few kilobases away from the target gene, but in certain cases they could be a staggering 1,000 kilobases away from the target gene. So just by similar to promoters, we can also categorize or predict the activity of enhancers just by looking at histone modifications around enhancers. And so here's the dashboard for the enhancer. So you can see the These are the histone marks that define inactive state of enhancers. And through a series of chromatin remodeling, these inactive enhancers can be turned into, or they can be activated into the active enhancer state. So just remember S3K4 monomethylation and K27 acetylation. These are kind of the hallmarks of active enhancers, and we'll discuss these later. This one is just a kind of summary of what we discussed so far. So you have 3 different chromatin states: active, bivalent, and inactive. Transcription start sites, they'll have both K4 trimethylation and K27 acetylation at active genes. Bivalent will have active as well as repressive mark, whereas inactive genes, they mostly contain K27 trimethylation and K9 trimethylation. And in terms of enhancers for the active genes, you will see the enhancers will have K4 monomethylation and K27 acetylation mark. And so now I would like to discuss some key papers. So this one is a really interesting example to show the power of ChIP-seq, how we can, using ChIP-seq and ATAC-seq, identify activation of enhancers that is driven by a specific transcription factor in this case. So ATAC-seq, you'll hear more about this technology tomorrow. So Thomas will be explaining in more detail, but just to give you a brief idea, ATAC-seq is an assay to give you a hint at the open chromatin in the genome. So wherever you have an open chromatin, if you treat them with hyperactive transposase TN5 loaded with adapters. So what this transposase does, it simultaneously cuts chromatin, the open chromatin, and inserts the adapter. So in one single step, you make library, and when you send these for sequencing and analyze the data sets, you will see these really nice peaks at open chromatin. Because transposase can't access closed chromatin, you wouldn't see anything there. So in this really interesting paper, what they have done is they have looked at the activation of enhancers when embryonic stem cells, they switch to epiblast-like cells. So just remember 2 different states of cells, and then the chromatin signatures, especially those of enhancers during this transformation. So they, in this paper, they identified this transcription factor, GRHL2. So these are all the sites where this particular transcription factor is binding during this transition. So in embryonic stem cells, there are not many open chromatin sites for these binding sites, but as soon as the transcription factor binds, based on the ATAC-seq heat plot, you can see there's much more open chromatin now. And at the same time, you can see the enhancers marks now coming onto these regions. So embryonic stem cells, there's nothing much in there. But in epiblast-like cells, you see K4 monomethylation levels are going up. Similarly, K27 acetylation mark, which is another active enhancer mark, it also goes up. And if you just make a average profile for these modifications and ATAC-seq data sets, you can see in the yellow, in the epiblast-like cells, there's a huge increase in ATAC-seq signal. Similarly, K4 monomethylation and K27 acetylation, so suggesting that these enhancers have been activated. And when you do the knockout of the transcription factor, in the green here, you don't see that activation. So it's a beautiful paper that shows how transcription factors mediate activation of enhancers during embryonic stem cell to epiblast-like cell transformation. This one is just a kind of a summary of what we discussed so far. I know it's a busy slide, but just remember there are 6 different states, chromatin states there. It's amazing that just by using histone modifications and a few other ChIP-seq data sets, we can distinctly define these 6 different chromatin states. And ATAC-seq, as it tells you, the open chromatin, so you would see enhancer and promoters. They have ATAC-seq peaks in there. And this one is the interesting bit, the poised state or the primed state. For the developmentally regulated genes, you see both active and repressive marks present there. Another interesting paper that I would like to discuss is this breakthrough paper in brain tumor biology. And what I really like about this paper is that they used motif analysis based on SVK27 estrylation mark, SVK27 ChIP-seq. And just by looking at the motif analysis, they predicted and they later on confirmed that SOX transcription factor, these are the main transcription factor that drive tumor cell initiation in brain. So what you see here is a profile for SVK27 estrylation ChIP-seq in cancer cells versus differentiated cells. So obviously you'll have some shared targets between these two different cell types. And there are some tumor specific and there are some differentiated cell specific peaks. So when these authors did a motif analysis on these peaks in tumor cells, they discovered enrichment for SOX transcription factors. So what it suggests that SOX transcription factors are most likely binding to these regions and kind of maintaining the transcriptional networks in brain tumors. So they further looked at the gene expression of all these neurodevelopmental transcription factors in cancer cells versus differentiated cells. So obviously all these transcription factors were expressed at really high levels in cancer cells, as also can be seen by SVK27 estrylation at promoter elements of these genes. And the most interesting experiment they did was they expressed all these 19 neurodevelopmental transcription factors in different combinations in differentiated cells and asked whether they could reprogram them into cancer cells. And just like Yamanaka factors, they eventually identified a set of 4 core neurodevelopmental transcription factors. So when you express these transcription factor in differentiated cells, they could successfully reprogram these into cancer cells. So that shows the power of ChIP-seq technology. So you started off with just SVK27 genome-wide ChIP-seq data sets. You identify the motifs, and based on these motifs, they could narrow it down to a few transcription factors and eventually define the core set of transcription factors. So everything seems really impressive about ChIP-seq, but the wet lab biologists, they could definitely relate to this picture because ChIP-seq is, it takes like forever to finish the experiments. So in an ideal world, when everything works really nice, right from cross-linking to the final libraries, it takes at least a week of pipetting and no one liked it, right? So the major update in ChIP-seq technology was this particular paper from 2015. And this method is called chipmentation. So what it did was actually reduce the overall hands-on time from 7 days to 2 days. It's much fast, more robust, as well as the best thing is that you could just do ChIP-seq with as little as 10,000 cells. So traditionally for ChIP-seq, we need 5 to 10 million cells. That's a lot for some experiments where you have limited samples, for example, embryos or biopsy samples or some other stuff. So the way chipmentation works is they use transposase TN5 transposase mediated adapter insertion, so similar to ATAC-seq. So the chip part is same as traditional ChIP-seq. So you start with cross-linked cells, sonicate your chromatin to 200, 600 base pair, and then go for chromatin immunoprecipitation. So while your chromatin fragments are still onto the beads, you add transposase. So what this transposase is doing is it's loaded with adapters. It binds to these fragments and inserts the adapter. So it's a one-step library preparation. So it brings down the overall protocol to just 2 days because there are like fewer steps of DNA purification, and the library preparation is much easier. So there's some cleanup after that, reverse cross-linking, amplifying libraries, and then send them for sequencing. Just one figure from the original paper, the chipmentation paper, just to show you the power of chipmentation, that it's highly reproducible and as robust as traditional ChIP-seq. So chip is ChIP-seq here, and CM is chipmentation. And what you're seeing here is snapshots from UCSC genome browser. So S3K4 trimethylation with traditional ChIP-seq, 500,000 cells. And when you do chipmentation, you see highly reproducible data, and you get identical peaks as with the traditional ChIP-seq. But the most interesting thing is that you could do chipmentation with just 10,000 cells, and you still can identify all these peaks. Same is true for transcription factors. Although with transcription factors, you need more input cells. So in this case, 100,000 cells compared to 10 million cells with the traditional ChIP-seq. So just with 100,000 cells, you can identify all the CTCF binding sites in this particular example shown here. And I can really vouch for this chipmentation method, because we have used this method. And in the last 6 weeks, I have generated more than 120 ChIP-seq libraries. So it's much, much faster, more robust. And especially when we are working with the embryonic samples, we have limited cells. So in our case, we could do ChIP-seq with just 20,000 cells using chipmentation. So it's a great method. So that's all about the ChIP-seq preparation and some of the key applications of ChIP-seq data sets. Let's talk a little bit about the fun part. So that's the ChIP-seq data analysis. And really the job of bioinformatician is no easier than the firefighters, especially if you are integrating ChIP-seq data sets with ATAC-seq and RNA-seq data sets. It's much more complicated. And one inside secret is that most of the time, the 90% of the bioinformatics is, installing, uninstalling different packages and kind of spending most of your time on Stack Overflow looking for answers. But anyway, tomorrow you don't have that problem, because we already have really nicely installed all the Dockers and the containers. But just to give you a flavor of what the overall pipeline for ChIP-seq analysis looks like, and you'll be using some of these tools and steps tomorrow. So for the paired end sequencing, you have these reads, the FASTQ sequencing files from the company, and then the first step is to do the quality control check. So you just want to make sure that you have enough reads and high quality reads. So FastQC is the preferred quality control check software. And once you have these peaks, you align them to the reference genome. These are two of the most popular and most efficient aligners. So BWA as well as BOTI2. So both of them work really well for ChIP-seq data sets. So once you align them onto the genome, you get these huge files called SAM files. So we compress them into binary files called BAM. Once you have the BAM files, you could use them for peak calling. So either using Max2 or GenRich, you call these peaks, and then you could use them later either for motif analysis or downstream analysis. Sorted BAM or the BAM files can also be converted to BigWig files for visualization on IGV or UCSC genome browser. So this is data from our own lab. So the input is kind of the background signal that you would see. And then after chromatin immunoprecipitation, you are enriching for K27 trimethylation mark here. So Hox cluster, it has lots of peaks here, as we would expect them in this particular case to be repressed. So it just shows you the basic outline of ChIP-seq data analysis. And finally, I would like to just briefly discuss this ChIP-seq data sets that you'll be using for the practical course tomorrow. So it's a really interesting data sets from published in Mall Cell in 2018. And what they have done in this paper is kind of looked at, they have tracked and examined these 4 different histone marks during early stage of embryonic development in zebrafish. So right from the gamete stage in sperm and oocyte, and then all the way to dome stage, and also kind of taking one control muscle cell, and they kind of tracked these histone marks. And then the most important and the most interesting theme that came out from this paper is that that SVK27 acetylation enhancer mark is kind of erased completely. So this process is called dememorization. And you'll be replicating this figure tomorrow. And what you see here is that sperm and oocyte with these two different marks, and also K27 acetylation for the later stages. So this particular enhancer mark, K27 acetylation, which is present in sperm, is kind of is erased, and in as early as 4-cell stage. So you can see these 3 different stages of the embryonic development. This particular acetylation, the enhancer, is kind of removed. So this process, dememorization, is also seen for the oocyte. So histone mark here, K27 acetylation is erased in the later stage. And this global eraser of the activation mark can be clearly seen here in the global heat map. So K27 acetylation in sperm as well as oocyte. So the yellow, the highlighted thing is the activated enhancers. And as early as in a 4-cell stage, these are completely erased. So philosophically speaking, the developing embryo doesn't learn anything from its parents. So it's kind of dememorizing everything and developing its own personality as far as SVK27 acetylation is mark is concerned. So good luck with the analysis of these data sets tomorrow. I'll stop here. Thanks very much for your kind attention. I have also included some of the papers that I really enjoyed. So some of these reviews. So they are a really good starting point if you have just started ChIP-seq experiment, either as a wet lab biologist or as a bioinformatician. Thank you very much.